Very good morning, one and all. Today we are discussing on endodontic biofilms. The most common etiology for the pulpal and periradicular pathologies is the microorganisms or the microflora. Infections in the oral cavity is caused by a number of organisms from different species found in the human mouth. These oral bacteria have the capacity to form biofilms on distinct surfaces ranging from hard to soft tissues. So biofilms means it's the group of microorganisms, group of bacteria. So the fundamental to maintain oral health and prevent dental caries, gingivitis, and periodontitis is to control the oral biofilms. Biofilm mode of growth is advantageous for microorganisms since they form three-dimensional structured communities with fluid channels. So this will help in the transport of substrate, waste products, and signal molecules. So biofilm formation in root canal is probably initiated sometimes after the first invasion of the pulp chamber by planktotic oral microorganisms after some tissue breakdown. So it was postulated by Svensater and Perinthalmanna in their study. Coming to the history, Anton von Leeuwenhoek in 1684 was the first to display animalcule, that is bacteria found in the plaque of teeth. In 1940, Peucelikian and Heller described the development of bacterial slime and colonial growth attached to surfaces. In 1943, Zobel described many of the fundamental characteristics of attached microbial communities in seawater. So, such communities were described as biofilms by Heramos, a Danish physicist, in the year 1978. In 1987, using the technique of correlative light and transmission electron microscopy, Nair presented the ultrastructural visualization of intracanal microbial flora and described embedded in extracellular matrix of bacterial origin and existing as sessile intricate communities promoting a symbiotic relationship, what is known as endodontic biofilms. So endodontic biofilm is defined as a mode of microbial growth where dynamic communities of interacting sessile cells are irreversibly attached to a solid substratum as well as each other and are embedded in a self-made matrix of extracellular polymeric substances. The bacterial biofilm should possess four characteristics, which are autopoiesis, homeostasis, synergy, communality. Autopoiesis means the biofilm must possess the ability to self-organize. Homeostasis means it should resist environmental perturbations. Synergy means must be more effective in association than in isolation. Communality means a response to environmental changes as a unit rather than single individuals. Endodontic microbiota transition is more conspicuous with the progression of infection. Nutritional and environmental status within the root canal changes as infection progresses. It creates more anaerobic environment and depletion of nutrition, which offer a tough ecological niche for the surviving microorganisms. The anatomical and geometrical complexities like delta isthmuses in the root canal favor the adhering of bacteria from cleaning and shaping procedures. Because biofilm is the manner of bacterial growth to survive unfavorable environmental and nutritional conditions, the root canal environment in both primary and post-treatment infections will favor biofilm formations. Additionally, biofilm mode of bacterial growth offers other advantages like resistance to antimicrobial agents, increase in the local concentration of nutrients, opportunity for genetic material exchange, ability to communicate between bacterial populations of same and or different species. So this ability of the bacteria to communicate between bacterial populations of same and or different species is known as quorum sensing. So it also helps in produce growth factors across species boundaries. Endodontic microbial biofilms can be categorized as intracanal biofilms, extraradicular biofilms, periapical biofilms, and biomaterial-centered infections. These biomaterial-centered infections is also called foreign body-centered biofilms. Moving to intracanal microbial biofilms. Intracanal biofilms are microbial biofilms formed on the root canal dentine of the infected tooth. So it consists of mainly loose collections of filaments, spirochetes, cocci, and rods. Apart from this, bacterial condensation were seen as palisade structures similar to dental plaque seen on tooth surfaces. In addition to this, extracellular matrix material of bacterial origin was also found in intracanal microbial biofilms. So this intracanal microbial biofilm is mainly composed of a microorganisms known as Enterococcus faecalis. So some experiments have revealed distinct stages in the development of E. faecalis biofilm on root canal tendon. So the stages include initially E. faecalis cell adhered and formed microcolonies on the root canal tendon surface. Later, they induced a bacterial mediated dissolution of mineral fraction from the dentin substrate. So this localized increase in the calcium and phosphate ions will promote mineralization of E. faecalis biofilms. So that means E. faecalis has the ability to form calcified biofilms on root canal dentine of infected tooth. Also, E. faecalis has the ability to co-aggregate with other bacterium like F. nucleatum. This co-aggregation interactions between E. faecalis and Fusobacterium nucleatum suggest the ability of these organisms to coexist in a microbial community and can contribute to endodontic infections. Interestingly, the calcification of bacterial biofilms and the bacterial mediated dentine degradation observed in vitro were supported by clinical evidences. Also, these biofilm formed by E. faecalis or the combination of E. faecalis and F. nucleatum can cause internal root resorption, periforaminal root resorption, and foraminal resorption. Moving to extraradicular biofilms. These are biofilms formed on the root surfaces. So, biofilms formed on the root surfaces adjacent to the root apex of endodontically infected teeth are called extraradicular biofilms. So, the biofilm contained varying degrees of extracellular matrix materials. The root surface biofilms are mostly multi-species in nature. The extracellular biofilm structures were dominated by cocci and short rods with cocci attached to the tooth substrate. Filamentous and fibrillar forms were also observed in the extraradicular biofilms. Some of the organisms like F. nucleatum, P. gingivalis, and T. forsythins were also found to be associated with the extraradicular biofilms. This is the SCM image of ultrastructure of extraradicular biofilms formed on the root surface adjacent to the apical foramen. So in this picture, we can see that a smooth structureless biofilm structure consisting of extracellular matrix material with embedded bacterial cells was noticed to coat the apex of the root tip adjacent to the apical foramen. Clinical evidence of calcified biofilm on the extraradicular region was also reported. So again, this bacterium can also form calcified biofilms on root surfaces. Ricusi et al. Has reported the presence of calculus-like deposits on the root apex of teeth extract post-treatment periapical periodontitis, while Hahn et al. Noticed calculus-like deposits on apical root surface of tooth presented with a lesion refractory to conventional root canal treatment. These calcified biofilms were associated with the periapical inflammation and delayed periapical healing in spite of adequate orthograde root canal treatment. Another category included periapical biofilms. Periapical biofilms means the periapical region of endodontically infected teeth are isolated biofilms which can be seen even in the absence of root canal infections. Periapical lesions which are associated with actinomyces species and propionibacterium propionicum can occur when the bacteria present in such biofilms overcome host defense mechanisms. So these actinomyces species in tissue grow in microscopic or macroscopic aggregates which may reach diameter of up to 3 to 4 mm. They are commonly referred to as sulfur granules because of the yellow granular appearance. This granular biofilm structure consisting of a central mass of intervened branching bacterial filaments held together by an extracellular matrix with the periapical radiating clubs. The aggregation of actinomyces cells influenced by pH, ionic strength, and cell concentration, which facilitate biofilm formation. It is important to note that the periapical region is patrolled by polymorpho-neutrophils and macrophages, which phagocytose incoming planktotic bacteria easily, but the phagocytes are unable to engulf bacteria in matrix enclosed biofilm structures. Say same image of the section the surface of sulfur granules showing rod-like bacteria, spiral form bacteria, and an amorphous material between the cells. Large amount of partly calcified extracellular materials are also present. Coming to foreign body-centered biofilm, also known as biomaterial-centered infection. Foreign body-centered biofilm is found when bacteria adhere to an artificial biomaterial surface and form biofilm structures. It is a major complication associated with the processes and also in implant-supported processes. Biomaterial-centered infection reveals opportunistic invasion by nosocomial organisms. So the common organisms found are Coagulus negative Staphylococcus, S. aureus, Enterococci, Streptococci, P. aeruginosa, and fungi. Studies have suggested that extraradicular microbial biofilms and biomaterial-centered biofilm are related to refractory periapical diseases. Again, bacterial adherence to a biomaterial surface is also described in 3 phases. Transport of bacteria to biomaterial surfaces, initially forms non-specific adhesion and finally forms specific adhesion between the bacteria. In endodontics, biofilm on root canal obturating material can be intraradicular or extraradicular, which depends on whether the obturating material is within the root canal space or it has extruded beyond the root apex. E. faecalis, Streptococcus sanguis, Streptococcus intermediate, Streptococcus pyogenes, Staphylococcus aureus form biofilm on GP points, whereas F. nucleatum, Propionibacterium, acne, P. gingivalis, and P. intermediate do not form biofilms on aperture points. The findings from the study suggest that gram-positive facultative anaerobes have the ability to colonize and form extracellular matrices on catapercha points, while serum plays a crucial role in biofilm formation. The catapercha surface was covered with the glycocalyx-like structures. Filaments, long rods, and spirochete-shaped bacteria were predominant in the biofilm formed on catapercha. Studies have suggested that extraradicular microbial biofilms and biomaterial-centered biofilms are related to refractory periapical diseases. So we will be continuing on method of eradication of biofilm on the following sessions.